thank you so much. I think we'll now open up questions to the audience. So I think if anyone has questions, you could create a single file line on this side.
Testing.
And Jazz will hand you a microphone. So feel free. You're telling me no one has questions? Okay.
Hi, my name is Anna. I'm a writer for PHR. Thank you all so much for coming to speak to us. Going back a little bit, all of you, I think, addressed the ethics and access issues with some of these therapies. And CAR T-cell therapies can range, as we know, some patients can face up to over $1 million for a CAR T-cell therapy. And you, Doctor, were touching on how now more than ever the government and the FDA has to take accountability in ensuring that the health insurance industry is caught up to covering these new treatments. So for both in the United States, in high-income countries, but also abroad in lower and middle-income countries, do you see potential avenues for making these treatments more affordable from a regulatory perspective, or is this has to be more from an economic or market perspective?
Well, I think it's both, really. Some things are getting cheaper. It's cheaper to make lentiviral vector. The CAR T cells are in the $300,000 to $400,000 range. There are associated potential toxicities with that. But when you compare it to the standard treatment, which is an allogeneic stem cell transplant, it is cost effective. Now, is it too costly to have every potential patient receive it? Yes. Is it too costly for South America and Africa and other regions of the world? Yes, and we do have to do better. I think we're making progress, but there does need to be some significant breakthrough, like the ability to use lipid nanoparticles. Now, there's another type of gene therapy that we haven't really talked about, which is for example, for spinal muscular atrophy. That uses a different type of virus, an adeno-associated virus. And the price tags for those therapies, the cheapest is $2.5 million and $3.5 million, and there's going to be one for $5 million. So there does need to be significant effort devoted to finding another vector or another way to deliver the gene to those different types of tissues.
Thank you so much.
Hi, my name is Eric. I also write for the Penn Healthcare Review. Thank you so much for your time. I really appreciated this discussion. I was also fascinated by our discussion about working with other countries. And so my question is, in your specific experiences working with other countries, like Dr. Weissman, you worked with Thailand, and Dr. Jun, you worked with China, Bioheng. I was curious about what are some political complications with licensing out intellectual property, particularly with the Chinese partner in Nanjing?
So we've attempted to work with multiple Chinese companies over the years, and the IP issue stands out as critical. This all started because when a company produces something in China, medical or any car, doesn't matter, they have to turn over all of the technology used to make whatever that is to China. And that takes away their safeguards of protection of their IP. So early in the pandemic, a Chinese company contacted me and wanted to make modified RNA vaccines. And if you look at China now, the vaccines they produced were terrible. They gave almost no protection, that lasted very little time, and they're constantly in lockdown. Penn wouldn't let me work with this Chinese company because of the lack of respect of IP. I didn't really care. I was thinking about patient lives, not IP protection. But at some point in time, China and other governments have to realize that this is going to be an impediment to improve that. There isn't much the US can do to say you have to respect IP rights.
I've been to China a few times, and on one visit to a new hospital, so new that the plastic was still all on the equipment, and they brought in people from a Chinese CAR T company, and the guy who was running their vector manufacturing facility came up to me and said, "Oh, it's so nice to meet you. We're copying everything you're doing." So, I mean, what do you say to that? And then how do you think about what a business relationship would be?
Yeah, I mean, so in addition to those concerns, which are really major issues, there's when we started in the space of CAR T cell, and the same thing with RNA, and it was only done here, and there were no trials in China. And now you can look on clinicaltrials.gov, and there's more CAR T cell trials in China than there are here. And their government has invested in this in addition to their venture areas. So they have a larger total part of GDP investment in cell and gene therapy in China than we do here. And I think that's a real, even without IP theft, it's a big concern. If they invest more, I worry that we could get pushed out of that area as far as innovation.
Thank you very much.
Hi, thank you so much for your time and coming here to speak for us. So my name is Yura. I'm a first-year bioengineering student, and I kind of have a general question about like biomedical research. But for those of us who are interested in learning more about the biomedical research field and cell and gene therapies, and for those of us who are relatively new to the field, like, what advice would you give to people who want to learn more about this field? Like, what can we do every day in order to keep up with the very fast-paced development in research?
Well, I'll give one answer is all the literature, newsletters, courses that you have access to. A selfish answer is I'm the immediate past president of the International Society for Cell and Gene Therapy. So join, because we have a early stage professional committee and opportunities to be mentored, to participate in the society, presenting your research and all of the resources. And it could be ISCT, it can be in another society, but network and get involved.
Thank you.
Yeah, I would just say be a sponge. And there are many societies that are trying to help foster development of, the new people entering the field. So it's a great time to come into this. The field's just beginning. That's the exciting thing. You're getting in at the ground level.
I mean, something I suggest to young people, to anybody, is give it a try. Volunteer in a lab for a month or two. Just experience what it's like to see, is this something I want to do? Does this really interest me? Because when you do that, you're going to learn about the technology, you're going to learn about the science, and if it excites you, then you can, apply to their grad school or do whatever you have to do to continue.
Thank you so much.
Hi, my name's Gina. I am a PhD student over at Drexel University. Thank you so much for your time. I have two questions. So the first one is around trafficking. So I was curious for all of your opinions in terms of the next steps for trafficking to difficult areas. You mentioned the brain being one of them. I'm also interested in solid tumors. You obviously have that very, cold immune environment. That's a hard hurdle to overcome. So I'm curious what the next steps are in terms of progressing that. And then my second question is more to Dr. Weissman. Are there next generation mRNA vaccines that are being developed to increase the durability of the immune responses following vaccination?
You have to be careful when you say durability, because our only experience is with COVID, and that isn't durability. The problem with COVID is you get a new variant every 3 to 4 months that's much less sensitive. So the only response we had was give more boosters to make more antibodies. we don't know what the durability of an RNA vaccine is for something that doesn't mutate constantly. So all vaccines have limited durability for levels of antibodies. The way vaccines work is they make memory cells that respond quickly the next time you see that pathogen. And those memory cells make antibodies and T cells that treat and block the infection. So I think with newer vaccines and RNAs, we'll learn what durability really is.
And to your first question, I think one approach to get trafficking, like with immune therapies, is to use orthogonal approaches. For instance, some people are using microwaves or ultrasound to guide cells into what would be a cold tumor. We have a trial that looks really promising now with a virus, an oncolytic virus that infects in the tumor of ovarian cancer patients, and then we give the CAR cells, and many more CAR T cells go in than would have if we hadn't used that virus. So it's combining two agents, and so there's a lot of work on this, and I think it's all going to be solved over the years. Yeah.
Great, thank you so much.
Hello, I'm Sai. I'm an early phase PhD student in Chris Bennett's lab. Thank you so much for your time today. I learned a lot from you guys. Just had two quick questions. One for Dr. Weissman. Earlier you said like when you know that something's good, you know. Can you elaborate on how did you know that, right? Because I have like maybe like 5-10 ideas every day, and then I'm like, this is it, but it's really not.
I think you have to go back to the wizard comment earlier on. Katie and I knew because we would sit down and we would list all the diseases that an RNA therapy could treat. And we would have pages and pages. So we saw the enormous potential. And other people's failures didn't stop us from trying to make it better. I mean, most of the things we do in medicine is trying to improve on what we did before. We used to do a lot of cabbages, open heart surgeries. Then they invented stents, so we moved to stents. And then they added drugs to stents. So all of those things are improvements. We thought we could improve the RNA and get it to work for these technologies. And with every bit of research we did, it was reinforced, so we kept going.
Yeah, my answer is a little bit different.
Yeah.
I started in immunology and adoptive cell therapy because I thought it was interesting. And that was at a time in the late 90s when other people and grant funders and meeting organizers didn't think it was so interesting. And it would be a session, as Carl says, on the last day in a room no one could find, and everyone there was your friend. And then things started happening, and especially with the CAR T cells having results that were too good to be believed, except they were true. So I think the first piece of advice is to do something that you enjoy, hope that it's sustainable, and if things get very, very lucky, really good things can happen.
Yeah, and my second question was more related to CAR T. Like kind of in hindsight, it's like such an elegant and really cool system. But when you guys were first starting it, what was the inspiration from like a science perspective to be like, let's put a T cell and engineer it and use it to attack our cancer?
It's a lot of ideas have, it's on the shoulders of previous ideas, and ours, there's like one of my graduate students made in his thesis a genealogy of CAR T cells, and on the top part, the problem with T cells in general is, my T cells won't work in you because we have different HLA genes, unless you're an identical twin. So that was solved by Zelig Eshar at the Weissman Institute in the early 1990s, when he grafted an antibody onto a T cell. And then there's another issue, and that's what Bruce Levine and in my lab solved, which is no one knew really how to efficiently grow T cells. And then the third was really solved by Richard Mulligan and Michelle Sadelain, where showing you could genetically modify T cells permanently, in their case with a mouse gamma retrovirus. So those things all came together then to efficiently grow and make CAR T cells. And there were different, but there was only 5 labs in the world working on it, and now there's thousands.
Got it. And why a T cell, not like a B cell or a macrophage?
Well, so people are working on B cells. I mean, you could install with some of Drew's technology, you could make an antibody, turn it into a plasma cell, and it will make antibodies as a factory in vivo. So that's and then, well, T cells we learned from the AIDS epidemic that they're the orchestra, they're a conductor for the entire immune system. And without T cells, the immune system falls apart. And we knew also, Bruce Levine did work early on when he was a postdoc with, where we studied telomerase, the enzyme that maintains telomere length. And we found that T cells were like the only cell other than really stem cells where you can turn on telomerase and they can live your whole life. So neutrophils that come out of our blood every day, your white blood cells, they last hours to days. Platelets last 5 days maybe. T cells last your whole life. Your thymus goes away when you're an adolescent, and then you live the rest of your life with those cells, and they divide, and it's a very coordinated way. So T cells are an ideal cell that way. You can genetically modify them, but they have stem cell-like properties.
Cool. Yeah. Thank you.
Hi, my name is Srijan. I'm a writer at PHR, and you recently mentioned in your answers about in vivo T cells, and specifically you could use that technology to reprogram, I guess, and mitigate Alzheimer's. How do you envision this delivery of medicine considering its difficulties, and how do you envision it will be in the future?
So we've got a number of therapies we're developing for neurodevelopmental and neurodegenerative diseases. We're also working on cancer therapies in the brain. They involve a variety of different targets. Sometimes for cancer, we target T cells. For neurodevelopmental, we target mutated genes in the cells of interest. The problem with the brain, so an LNP is an 80 nanometer particle. It's the size of a virus. The blood-brain barrier does not want them to cross and does not allow them to cross. So you can stick LNPs in somebody's brain. It's not the satisfying thing you want it to be. So we're working on giving them intravenously, getting them to cross the blood-brain barrier, which we've done. And now we're delivering a variety of gene therapies and therapeutics. The brain therapies are 5 years behind everything else because A, it's incredibly difficult to work with, and B, the pathophysiology, the gene defects are only now becoming investigated, whereas years ago we knew about immune deficiencies and cancer genes and many other genes. So I think neurotherapies will be developed over the next 5 to 10 years.
Thank you.
Hi, thank you guys for being here. My name's Natalie, and I work in genetic counseling over at Penn. So in genetic counseling, we have a big issue with insurance companies covering diagnostic tools like whole genome sequencing. So when this inevitably becomes clinical, how do you expect to encourage insurance companies to cover a therapy that might not even take the first time?
Yeah, I think that insurance coverage is a significant issue. I've heard of CAR T cell patients for coverage. The carriers are putting preconditions on, like you have to get a colonoscopy, you have to do this, you have to do that. On the other hand, I've heard from Steve Grupp at CHOP, who's been running the Vertex exocell clinical trials. Actually, the one that was just approved that's being covered so far, which is great because I think this year we're going to have another sickle cell drug approved. That's the XSL. And that's a CRISPR modified stem cell. So I think that's a milestone for two reasons. First, approved CRISPR product, and also in a diverse population. But I think there does have to be some additional pressure, maybe some initiative, and it's got to come from the patient voice, so the people that you're speaking with, and for everyone else to be able to amplify that. But the companies, I am a Penn alum, and I had roommates from Wharton. I say not a lot rubbed off, but maybe some by decades later. But that is a policy and a business issue from a poorly designed or designed to oppress healthcare reimbursement system that we've all got to work together to solve.
For sure. Thank you.
Hello, good evening, professors. I'm curious, as in based on your various experiences, how important do you think it is for future bioengineers and biomedical researchers to be knowledgeable in the business domain, like business administration, market analysis, so that once their technologies get past the clinical trials, it could like reach and benefit a greater population?
Yes, or you need to know someone who knows that stuff. And I think that this is something we haven't touched on in conducting translational research or from discovery to clinical trials is having teams of teams. So we all can't be an expert in everything. And in the center that Carl directs, we have 25 research labs. We have a scale-up product development laboratory. We have a manufacturing, we have QC, we have QA, we have regulatory, we have business operations. So everyone has a role and is an expert in something. And then everyone should know the person to ask for expertise in an area where they're not an expert.